Hi, everyone. Welcome to the research updates with Dr. Cody and Dr. Hale. We hope you're enjoying our conference so far. I'm going to now pass it over to Dr. Cody, and she will get started. If you have any questions during the presentations, go ahead and type them into the chat box, and we will get to them at the end. So welcome, Dr. Cody.
Hi, everybody. Glad you could be here. I usually think of myself as the most technologically challenged person there is, but we've just proven that actually that distinction is for Dr. Hale. So he's working to get on the video at the moment. And he was going to do the beginning of this talk, so I'll start and let him jump in whenever. One thing, you probably all figured this out already, but I did notice that if I go to the full screen, I can see the slides better, but then I can't see the chat. So in the other talks I've watched, I've gone back and forth between full screen and chat, just so I can keep track of what people are saying, but I still really want to see the whole screen. So welcome to the research update. Let's see, first, whoops, am I going the right direction? My slides don't want to advance. Oh, I know why. Just wait a minute. Yay, sooner or later I figured it out. First, most importantly, our disclaimers, where you don't have any actual or potential conflicts of interest related to the presentation, and we don't discuss unapproved uses of commercial products or devices. I am employed by the University of Texas Health at San Antonio, and Dr. Hale is employed by Penn State Milton S. Hershey Medical Center. So there. So the chromosome 18 conditions, with regard to the registry, I think you probably, all of you already know there's a huge amount of variability from one condition to the other, but we put everybody into one of these 5 buckets, 18Q deletions, in which someone can have a deletion of, I wonder, can you see my cursor? Does that show? Oh, it does show up. Oh, wonderful. Okay. Someone can have a deletion of any size from any portion of the long arm, and it's 18Q minus. So as you can imagine, there's a lot of variety, and not necessarily, they're not all overlapping. By the same token, you can have a deletion of 18P of any size of any region of the short arm, and it's 18P minus. Ring 18 involves a deletion of both ends of the chromosome, and so each end can have a different size deletion. In these cases, it's always off the end of the chromosome, so there's huge variety there as well. And then there's tetrasomy 18P, in which individuals have an extra chromosome, but that chromosome's made up of 2 P arms. And so, where'd my cursor go? Okay, so you have 1, 2, 3, 4 P arms, so tetrasomy 18P. And almost everybody with tetrasomy 18P has the same thing at the molecular level. Only a couple people have had Q arm duplications as well, and they had extra medical problems because of that. So that's our most uniform group. And then, of course, there's trisomy 18, which by far is the most common. But people with trisomy 18 are very medically fragile when they're babies. And most do not survive to see their first birthday. And so the families with children with trisomy 18 have very different concerns than all the rest of the families. And so there is another group that existed before us called SOFT, Support Organization for Trisomy 18 and 13. And most families belong to SOFT, as well as the registry. Either way, you can belong to one or both. We don't care. We're sister organizations. We hope to be able to fill whatever needs you have regarding the syndromes. And our goal, of course, is to make the chromosome 18 conditions the first treatable chromosome abnormalities. And by that, we mean self-sufficiency. When we go to Washington, we tell the people on Capitol Hill, we want to make our kids ineligible for SSI. Wouldn't that be fabulous, actually? And we don't want to have special needs trusts. That's our goal, is to have the same sort of expectations for our kids with chromosome 18 abnormalities as we have for our typically, or sometimes not so typically, developing children. So that's going to involve establishing therapies employing established therapies, identifying what those might be, and creating novel therapies just for people with a specific chromosome 18 deletion or duplication. So that's our goal. And the approach is completely different from most rare disease disorders. So if you have friends who have a child with a rare disease, which many of us do because that's the world we find ourselves in special ed and et cetera, and Special Olympics, and those friends have kids with rare conditions, and they're involved in support groups who are interested in treatment and research, and you see the kind of path they're taking. And I mean, way back when, everybody was all about, let's identify the gene, we'll have it made. We already knew where all our genes were. We just didn't know what they did. And so the conditions of being a chromosome abnormality completely defines how we approach these, and it's different from everybody else. Now, what we do have in common with other chromosome abnormalities is that these are rare. In fact, they're not just rare, they're ultra rare. In fact, here are the numbers if you want to jot those down quickly. 18P and 18Q occur in about 1 in 55, 1 in 66,000 births. These are just estimates based on data gathered before microarrays were typically done in the clinic. So they're probably a lot more common than that, but even if it's twice as much more common, it's still ultra rare. Ring 18 is 1 in 300,000 births, and tetrasomy 18P 1 in 625,000 births. Now that is ultra rare. And as you can see, trisomy 18 is not quite so rare, 1 in 7,000 births. So if you think about it, I often get people saying things like, well, my doctor didn't know anything about it, or why isn't there a clinic for this in my town? It's a big city. Well, this is why. First of all, if you combine all the non-trisomy 18 conditions, they occur in 1 in 24,000 births. So if you think about it, an average pediatrician might have 300 patients in their practice. You know, you'd have to be talking to a whole room of 80 of them in order for one of them statistically to have seen one person with any one of the chromosome 18 conditions. So they're pretty rare. And in comparison, for every one person with one of the non-trisomy 18 chromosome 18 conditions, there's 35 people with Down syndrome. That's enough to have a clinic. One person isn't enough to have a clinic. There's 6 people with cystic fibrosis and 2 with Angelman, another rare disease. So, so now you understand how we compare number-wise to some of these other conditions that you thought might be also rare conditions. Our solution for that is to have one single coordinating center. Try to keep all the information in one place, not scattered around 4 different universities that have to collaborate, fight for funding, and try to coordinate all the activities, or argue among themselves and not share information. That would be really disastrous. The next challenge that we have that's defined by these conditions is the variety in size and location of the chromosome 18 change, and which I've really already talked about. Here's just one example. Somebody with 18q minus might have a big deletion, a little bit smaller deletion, a little bit smaller, or even a really small deletion, and these are all terminal deletions from the end of the chromosome, or they could have an interstitial deletion here or here or here. These could be any size, any type, and so there a huge variety in the molecular cause, the underpinnings of the condition, and consequently the kids are very, very different, or adults are very, very different in their abilities and their challenges. Likewise, with 18p minus, half the individuals have a deletion of the entire P arm, but the other half have a unique deletion. So that means we can't really do a syndrome description for P minus or Q minus, for example, or ring, because there's so many different. Whose deletion do we pick? And that really goes for the duplications of, because we have members with partial duplications of 18p and 18q. So our strategy then is to focus on the genes. By understanding the role of each gene, then hopefully we can begin to understand the role as they apply to any one person. So we could say, okay, your child is missing these 10 genes, and these 8, that doesn't matter when you have a deletion of those 8 genes. This one explains why they have this condition, and this one puts them at risk for something that we need to watch for so we can manage it early. So that's why our focus on the genes. We also have, of course, genetic complexity and outcome complexity, which I think you've probably already figured out. And the way that affects us is that on a single gene disorder, there's usually one major organ that's affected, and there's one set of biological processes or anatomical developmental processes that are affected. So you have a whole bevy of clinicians who specialize in the function of that organ, and you have a whole herd of scientists who specialize in different aspects of the developmental or biochemical process. And so it's really easy to find out who those people are across the country and get them all together. We have multiple organs affected. In some ways, we don't exactly know how at a real basic biochemical or developmental level. Then we have all these 263 genes, and we don't know what they all do. And so we have a big challenge here. And so even when we start to figure out which genes are important for which things, we often have one organ that might be affected by 2 different genes, maybe causing 2 different problems, or working together to cause a unique problem. We could have one gene affecting 2 completely different organs that you might not think would be affected by the same gene. And then, of course, we have organs, specifically the brain, that are affected by many, many of the genes that are on chromosome 18. So we have this challenge of genetic complexity. And so our approach because we're taking a gene by gene approach, we want to identify the genes that are dosage sensitive. They're sensitive to the dose or the number of copies of that gene, which is not really all the genes. And if you've heard us talk before, we said 15 years ago or more that we thought it was going to be 10%. And as we are learning more and more about the genes and what they do, amazingly, that hypothesis has really borne out. And really, it's only about 10% of the genes that have an effect when you have an abnormal gene dosage, meaning an abnormal number of copies of that gene. Then, of course, and I just was looking at my table on my desk, and this is slightly off because some of these have changed. But if you look at the 263 genes on chromosome 18, only 17, and now I think I've modified it a little. Let me look at my list here. Up to 15 that are actually causal, that we think cause a problem in more than 50% of people with a deletion of that gene. 9 that are low penetrant, so less than 50% of the people have a problem caused by that gene when there's a deletion. 49 are conditional, meaning and another 15 risk factor. Those mean other genetic conditions have to be present for that to cause a problem. So it's sort of no effect, but when somebody has something unusual that we don't usually see with a deletion of this region or of this specific gene, we have this list of things that they're probably a little more at risk for than the general public. So that's why we keep track of those. But you can see it's a small number. With the duplications, by the mere fact of a duplication, there's fewer ways to interrogate that scientifically, but we're working on it. So for duplications, the number of unknown is a lot bigger than 25, like I'm showing here. So this is just an example, but we do keep track of that all the time and have things we'd like to do to investigate it if we could just afford to do that. So we also have outcome complexity. And so, whoops, let me go back. And you talk about that a little more. By that, as you've probably figured out from the previous few slides, we're trying to parse that out and figure out exactly if, for example, intellectual disability, for example, being the most complex, we're trying to understand, is it working memory? Is it speed of processing? I mean, what are all the components that make that up, that people with chromosome 18 changes are more likely to have or not have than somebody else? So extensive characterization. And then when we start to understand those characterizations, delve into that even further and find out the basis for that, because the more you look, the more subcategories there are, and so we can keep delving into the underlying processes. And our goal then is to try to get to a measure that's as close to the biological process that's not going right as possible. And that's for two reasons. One reason is it may inform the cause, and we may give us insights into therapies or other existing treatments. But the other reason is the closer we can get to understanding the biological cause, the closer we can get to being able to devise a novel therapy. So either way, that's important. It gets us closer to our goal of being able to make these the first treatable chromosome abnormalities. So our approach, taking all that into consideration then, as I said, is to establish a single clinical research center. And we've really focused on the problems directly affecting the families. We really haven't worried about the molecular mechanisms of duplications or non-disjunction or whatever it is. It doesn't really matter. This happens in most cases as a random process. So understanding that's not going to help our families. Our families have people, loved ones with chromosome 18 abnormalities. That's a given, and that's what we're going to focus on, what can help them. This means that families have a single point of contact. You should not be approached by multiple investigators, one wanting to study this aspect of a neurological condition and another one also wanting to study the same thing. And so we don't have competing things. That's what we do here, is we sort out the most appropriate investigations with the best possible investigators to collaborate with. We also, of course, do comprehensive clinical evaluations. We haven't been able to do those in the last few years for lack of funding for that, but you'll see in the last slide, some of my dream things that I really want to be able to do. And so we hope to continue those. It also means when all the data is in one place, we have a consistent set of information and a critical mass of information all in one place about the chromosome 18 conditions. And I can tell you there haven't been hordes of people asking to help people with chromosome 18 abnormalities. It's ultra rare. And when I see papers that are review articles on chromosome abnormalities, they hardly ever mention chromosome 18. They measure or mention the ones that are more common. And the team who is dedicated to this goal of making life better for people with chromosome 18 abnormalities really is a long list of people. Many, if you've been here or you've been to other conferences, you know a lot of these people, you've heard them speak. And so they're from all kinds of medical specialties, endocrinology, psychiatry, neurology, ENT. And as we've learned more about specific problems, we've added people with Dr. Morer with expertise in developmental bone health, Dr. Meyer with expertise in platelet function. Also, as we've learned more and we're starting to understand which are the important genes, we're looking at mouse models and cellular models. So we've added investigators who are expert at mouse behavior, mouse electrophysiology, developing, creating mouse models, and then cell-based assays. I'll talk more about that later. So the team is growing, actually, as we've learned more. Our current enrollment numbers, which were significantly slowed down by the pandemic as all our lives were changed fundamentally. But you can see we have an amazing number. And when I talk to people about our cohort, and I say we have nearly, actually by today, I think we do have 700 people enrolled, they just look at me in shock and can't believe we've got this huge number in our cohort. And so you can see at the research center, we break out trisomy 18 into people who have duplications of 18q or duplications of 18p. So we have a few more categories than the registry uses. And of course, they come from all over the world, and mostly North America and mostly United States for obvious reasons. But the research study is really a lifetime longitudinal study. So if you're involved in the study, you get surveys from us asking about bone health or autoimmune disorders, allergies or seizures. We ask for medical records all the time. Anytime you have a new medical development, we want to know about it. Send us your medical records. And so that involves lots of back and forth. And while we could easily translate our consent forms, because anytime you're involved in research, you need to sign consent forms explaining all the what the research is, what it's about, and what the ramifications are. Anytime you sign consent forms, we could easily make them translated into other languages, but that's not the end of the story. We go back and forth, and so we don't speak lots of languages. There are people here who speak Spanish, but beyond that, not so much. So speaking English, and also I should mention, some of the surveys that we do are validated, especially behavioral, neuropsychological kinds of surveys. And those instruments are really only validated in English. And so that means our data can only be done in English. So that's definitely a limiting factor. The other limiting factor is we need to get blood in order for you to be fully enrolled, and we need to get blood samples within about 3 days. So those are our limitations. But given that, we're still able to enroll people from all over the world, which we're always very excited to do. We love to be able to add new countries to our list. So our strategy then, of course, is to devise new treatments or to identify standard treatments. And by that, I want to make sure that you understand that we really think about this very broadly. And by that, of course, we mean typical drug treatments, but also it could be exposures. It could be an environmental exposure that we learn that you're more at risk for having an effect, adverse effect, if you're exposed to a chemical or something, or any sort of exposure. If we learn that, that's going to be important for improving people's lives. So I consider that a treatment. Dietary treatments could be appropriate to either avoid something that is harmful if you take in too much, if you have a specific chromosome 18 change, or it could be supplements that you need, either food or just a typical dietary supplement in a pill form that you need to optimize your health. We don't know about that yet, but I would consider that a treatment. So we think about this broadly. And of course, there's experiential treatments. For example, Liz greatly benefited by doing Fast Forward, which is really is aimed at people with dyslexia. Not sure that she actually had that, but she was tremendously helped by that. It changed her reading level by multiple grades in a couple of weeks. And that was a therapy based actually on real data, not just somebody's pet. They really think it helps kind of experience, but really validated therapy. So we think about therapies very, very broadly. And let's see. And to get to that, we kind of have two approaches. One, you can think about it as top down or people first, whichever. So I think of it as top down, people to the genes. And I've kind of already explained this, that we want to understand all the characteristics of people with chromosome 18 abnormalities so that we can employ the best treatments, but also that helps us understand which genes might be the important genes. And even if there is a treatment for some aspect, when we know what genes are involved, that may give us insight into what might be a very much more specific treatment, less likely to have side effects. So I think that's really been our major approach, is to understand all the characteristics and try to understand how we can help people best with chromosome 18 abnormalities by really understanding the characteristics of the people. I think I just said this, didn't I? By investigating the clinical features. Oh, and the reason I put this on is so I don't forget that we're interested in the congenital aspects, I mean, the things they're born with, but also late onset conditions. Nothing is really known about adults with chromosome 18 abnormalities. We don't know, are they at higher risk for some adult Parkinson's or Alzheimer's or some other adult onset condition? We have no idea. That's why this is a lifetime longitudinal study, because we want to know about that. So those are the kinds of things we're following. And if we find out that there is such a condition, then we're going to start monitoring for it. We also, of course, use this information to create the management guides, which I hope every single one of you has seen. They're on the website for the Research Center. And that's where all this information from our 60 plus peer-reviewed publications has generated, the knowledge in those publications has generated the information that's in the management guides. And that's just the same information repackaged in a way to be more useful for clinicians. So an example of the top-down strategy is our early observation that they have poor growth. People with 18q deletions are very often small. Sometimes it's called failure to thrive. Sometimes it's just short. But we learned that it's caused by a growth hormone deficiency in most cases, and that's a treatable condition. And so that's our example we use because that was our early finding. But anyway, but that's our overall strategy, and we continue to work on this. The other strategy is bottom up, to learn about the genes and then say, oh my gosh, this is a gene that could affect someone with a chromosome 18 deletion or duplication, and then follow that up. So by identifying the key genes and defining and understanding how they act, what they do, then we want to learn how to regulate that. So an example is the gene called SMCHD1 was found to be responsible when you have a mutation that causes one copy to be non-functional, which is really analogous to having a deletion of one copy. It was found to be associated with facioscapulohumeral dystrophy type 2. Whew. Took me a long time to learn how to spell that, but FSHD2 is usually adult onset type of muscular dystrophy. And we were very concerned that people with 18p deletions were very at risk, high risk for this. We collaborated with really a worldwide coalition headed by a group in the Netherlands, and determined that the molecular mechanism of this is really complicated, involves more genes than just the gene on 18p, and that people with 18p deletions are not really at a huge risk, but maybe a small risk of having this. But it's adult onset. Our cohort's pretty young. So we continue to try to follow people, and we have a study where we're asking people to do video interviews on an iPad which are evaluated by the neurologist, who start to see if they can see first signs of this condition. So this is where the genes inform the clinical assessment that we want to do. Again, all focused on treatment and identifying ways we can make life better. So the process, just so you have an idea of how things work, they all begin with an idea. That idea may come from knowledge of the gene. It may come up from a clinical investigation that we when people come here, that something that we identify. It might come and does come from you. Families tell us, oh my gosh, my kid has this and there's 3 other kids with that. And we say we didn't necessarily know that before. So what do? We do a survey. We ask the families with that same condition. Or we do a clinical assessment. We ask people, go to your physician. We have a concern about this. Be evaluated for this sort of thing. Somehow we do an investigation. It might be in the lab. We look at something in our cell lines or in the DNA specifically. Anyway, so we do some sort of investigation. That's the next step. And of course, then we have to analyze the data. That can take some time. Sometimes it requires collaborating with some experts to help give us some information about that and how to think about it. And then, most importantly, we disseminate that information. And let me tell you, those people who are enrolled in the research study, they learn about it first. The first thing we do is we let the participants know, what did we find? And usually all the data is finalized, and this is sort of a timing thing. We submit the data for publication in a medical journal. Here's a cover off one of them with pictures taken by Rick Guidotti. This was actually an issue about chromosome deletion syndromes that we were editors of and had several articles in. But this process of getting something published can take 2 years, really take 2 years. So what do? So first, we let the families involved in the research know what's happening. So they always get first take on everything. They learn about it first. If it's something, a really new finding, we put it in the management guides right away. And usually I put it in this talk right away. All that going on probably while it's all being peer reviewed and in this process for getting published. In our case, peer review has never really changed the finding. They change how we might talk about it or something like that, or say add more information about this, but it doesn't really change the finding. And so, like I said, the first people who hear about it are you all. The very first people are participants. Usually after that is the registry family altogether. I usually do a video or there's a little blurb in the newsletter, the email newsletter. And it goes into then the management guides. If it's really fundamentally, if it's a new finding, it'll go into the management guides. And then it gets published where anyone in the whole world can access it. So, so that's the process. You hear about it first. I want to make sure you know that. But it's a process and it takes a long time. And writing up a paper takes up a tremendous amount of time. And back and forth, and make sure all the authors are happy with it. And so it's a very long process, but essential. So I still haven't seen Dr. Hale come on, and I'm way past. He was going to talk first, and then I was going to talk with the last half, and I haven't seen him. Oh, hi, Holly. Did we totally lose Dr. Hale? I usually talk over him anyway. Oh.
That's okay. Once you're finished, you have some questions. So
okay. So I'm not quite finished yet because I wanted to talk about the pandemic. And I mean, this has disrupted all our lives in all kinds of ways. And while we have continued to work, the Health Science Center, we're a medical school attached to a hospital where we are an essential business. I have a private office here. I came into work every day. I never stayed home just because I have 2 monitors here. All my stuff is in here. You can see I got it decorated the way I like it. I'm home here. So I came in every day, but I was here all by myself, door closed, all by myself most days. But other than that, this has had a tremendous impact on what we can do, the rate we can do it, and whoops, oh, I know, I was going to tell you a story here. So what really happened when the pandemic hit? One of my other sort of jobs, which really didn't take any time, this is a no thanks to Dr. Hale. He and I were co-directors of the Pediatric Clinical Research Unit for all the research that happens in the Department of Pediatrics here. And of course, when he left San Antonio, that sort of left me as director. But was very lucky. He hired a brilliant, hardworking, amazing coordinator, project manager for that, who ran that unit, which meant I really didn't have to do a whole lot. Didn't take up a lot of my time. And so when the pandemic hit, all the clinical projects in the department, they just stopped because nobody was in clinic for a little while. But the adult ID, infectious disease service, suddenly had treatment trials that were that they were a part of, that were across the whole country. The remdesivir is where it started. Surely you've heard of that as a treatment for COVID. And the infectious disease docs said to some higher muckety-mucks here at the Health Science Center, who's the best coordinator on campus? We got this project that's starting like yesterday, and it's giant, and we need help. Who's the best coordinator on campus? And of course, it was our coordinator. And they asked her if she would help, and she said, sure. She's part of our group. She's in office suite right across the hall. She's in the same office suite with Bridgette. She was immediately overwhelmed with work. And she's part of our group, a kind of our extended family. And so what happened? Bridgette, who's in the same office suite, jumped right in to help. So Bridgette immediately was gone as of, I think, June of last year. And since she has decided to stay with the remdesivir study because she really likes that project and what they're doing. And she's going to kind of stay on with a few percent time, maybe in the fall, if she can squeeze it in. We'll see. Trying to maintain what's going on and not totally lose it. And as you know, she's our psychometrist, so she does all the neuropsych kind of surveys that we do. Also, completely unrelated to the pandemic, Courtney Siebold, our amazing genetic counselor, is moving to Sofia, Bulgaria. And they're in the middle of their move right now. Her husband has an embassy job, and that's just not going to let her, with their 2 young sons, be able to stay a part of our staff. And she's going to move to a kind of a volunteer position, which I'll talk about in a little bit. So Anna, thank God, is not going anywhere. So our staff is a little bit smaller due to the pandemic, but Annis is still here, and I'll give you her contact information later. The other part of our research staff, Patty Hurd, who runs the lab, as I said, the pandemic hit and our coordinator went off to do remdesivir, and Bridgette jumped in to help, and Patty jumped in too. Part of the reason, twofold. One, she's meticulous, data-oriented. Thank God for that. It's an essential part of her job. But because of the pandemic, we didn't think people would be sending in blood samples, so we wouldn't be enrolling people. So people had fewer opportunity to go to the doctor and get blood drawn, and probably didn't need to do it for something totally optional like this. So we put blood samples on hold, and Patty also left in June. She's now back at part-time working in the lab, and part-time still on the remdesivir studies. And Mimi Hassi, who is very quiet. You may never have met her, even at the conference, even though she's always there. She had to resign just recently due to health reasons, so completely unrelated. So now we have Patty part-time and David Rupert, who's our data manager, who is still with us. So our ability to move forward has very much slowed down due to the pandemic and other extenuating circumstances. So this gives us a chance to regroup. Oh, there's Dr. Hale, and as usual, I never let him talk, but I'll let you talk in a minute, okay? And you can fill in all the things I skipped. And I can go back to slides if you want, Daniel. So all of this has given us an opportunity to regroup and rethink, and what are we going to do? And you know, I'm not getting any younger, and so it's time to think about bringing on an associate director. So we've asked John Gelfand to come on in that role in September. In September, it's only going to be 10% of his time. He's on lots of projects. Many of you may have already met him. He's always at our conference for most of it when it's here in San Antonio. He sits quietly in the back. He really does. He gets it. And to me, that's one of the main reasons. And in fact, and the entire team just loves him. He's a great person, easy to work with. Even though it does say biostats on here, he's easy to work with. He went to medical school so he could understand the biology behind what he wanted to study. He's not licensed to practice medicine, but he knows all the medical terms and all the things a physician would understand about medicine. He's actually been a part of our team since 2008. And really only in the last couple of years, he has not been had part of his salary covered by us. We haven't had the funding to do that for the last few years, but we had kept him on at a small amount as long as we possibly could. And he actually even had a training grant when he first came on faculty here in 2008, I think it was, maybe 2007, to do some of the chromosome 18 work. So you'll all be getting to know him better in the near future. I might actually let him talk, except Dr. Hale will tell you I hardly ever let him talk. So that's one new thing. And the other part of that is because of the pandemic, funding, you know, the registry, it uses event-based funding. We have a golf tournament coming up. Power of 18 this year didn't do so well. It's not over yet. There's still a chance for all of you. Our funding is very much in question going forward. We've been able to maintain so far, but it's hard. I can't justify with my chairman that we can hire somebody new when funding in my mind is all full of question marks, and I'm scared to death. Until some individual fundraisers start picking up, people start being sponsors for the golf tournament, I can't start replacing staff. And so until then, we're working on all the same things. It just almost everything lands on my desk, and there's just only so many hours in a day. And so I'm here every day and working away as fast as I possibly can. So let me tell you a little bit about another new development. This is a registry development, very important distinction. We have started a scientific medical advisory board. These are volunteers. This is a registry board, advisory board, because it's critical that the Chromosome 18 registry, as the nonprofit, as the voice of the families, sort of direct what's happening at the research center. I am that voice. It's one of the 2 hats or 3 hats that I wear. Made sure that the patient family voice stays the focus of the research. Someday, I'll sort of retire maybe someday. But in preparation, we need to start having an outside the research center advisory board, helping the registry to direct the activities. And I'm trained as a geneticist. As we move to therapies, I'm not a biotech person. It will require expertise I don't have. So, and John Gelfand will be on this in this group. Most of these people, I think you know, most of you know. Of course, Dr. Hale. Steve Hobby is one of our dads, and he's a pediatrician. Bob Tavernier is also one of our dads. He works for Blue Cross Blue Shield. When we start thinking about therapies, we got to think about insurance. So he'll be a good voice to be involved. Brian Westman is also a parent. He's an ER doc and an anesthesiologist at WashU. John Carey, I hope some of you have met and know. He's really Mr. Trisomy 18. He was involved in the World Congress, so he was at that in 2004. He was at our first Utah conference, our second Utah conference. He unfortunately was, I think, out of the country at the time. But he has been a great friend and collaborator to us from day one. So he very much knows all that we've been doing over the years and what our goals are. And then Catherine Larson, whose talk I just listened to. I hope you just listened to it. My daughter is a child psychiatrist. So since we're really focused on obstacles to independence, behavioral aspects are really important. Courtney Siebold is joining this group as a volunteer. She obviously knows a ton about all that we do and all of you. And John Gelfond will be joining the group as well. Just to highlight the projects we're working on, recently completed projects, we did a behavioral issues study with Jonathan Mason, who's going to be talking. I think it's next. He's next. He's this afternoon. He's from Sunshine Coast University. We met him when we were in Australia. He really was excited about chromosome 18 conditions and wanted to do a study. So one of the beauties of doing this virtually is that we get to have him talk about his study. There's a publication coming out soon. It's actually study's been submitted for publication on the mouse model for a gene on 18q. It may have a minor effect on some of the congenital malformations. So we're interested in looking at that some more, but we were involved in that and helping them analyze that data. And this year we also completed a trisomy 18p management guide, mostly based on the literature because we don't have very many participants with that. But those are things completed this year, so we really were able to keep moving it forward. We're working on analyzing the allergy immunology survey data, and we just closed a trisomy 18 long-term survivor survey of people with over a year of age with trisomy 18. So that's just closed, so we're just starting that analysis. Active studies, we just submitted, we are, I think we're in. It's the NIH, okay? So we have been accepted, we think, into a project to do whole genome sequencing on the members of our cohort who have, they don't really care which chromosome 18 condition. They want people with structural birth defects. So that's their criteria. And all of these people need to be reconsented because really the beauty of this project is that they will do, no money comes to us, they will fund a sequencing center to do a whole genome sequencing on our participants with structural birth defects. And we have, when that data is done, we have 6 months to analyze it ourselves. And after that 6 months, it becomes public. But by that, by consent form language, it's public, but it's not really public. Investigators, any investigator who gets approved by the National Institutes of Health as a valid, real investigator with a valid reason to analyze the data will be given permission to analyze our data and the other data in this this is called the Gabriella Miller Kids First Pediatric Research Program, any other data in that program. And what that does is two things. I mean, the plus is it opens it up to other people outside of us who might have really good ways to analyze the data, really interesting insights that we never thought of. So it grows the cohort of people who are helping us learn more about chromosome 18 abnormalities. But it does involve having to have everyone reconsented. So If you have been asked and you ignored the emails from Anna's about being reconsented, please send the consent forms back because it is a broader use of data than we've had before. These people will never know your name. They may come back and say, we found this variant in this gene. They'll have to ask us. Does this person have any condition related to this variant? We can look it up in our database. We could ask you. They will never ask you. No one else will know who you are. So they'll have to come back to us as the coordinating center. So I think it's a huge leap forward for us. It's a huge opportunity to get other people involved in chromosome 18 conditions. So we're really excited about that. We also just sent in a grant about 3 weeks ago, trying to determine which genes cause myelination problems in 18q. It involves creating human brain organoids from cells from someone with an 18q, or multiple cell lines from different people with 18q deletions, and also looking at mouse cells with deletions of those genes, ultimately to create a mouse model. That grant was submitted about 3 weeks ago. It will get reviewed in October. If funded, great big if, because only 10% get funded, then it would start in April. So that's the National Institutes of Health timeline. Not fast, but those are the active projects I spent a lot of time on we've all spent a lot of time working on in the last year. The other active projects, as I've already said, the video assessments for first signs of this type of muscular dystrophy, and also we're looking at speech at the same time. 2 different sets of investigators look at those videos. We're also looking at wearable devices, kind of Fitbit kind of devices that can download data that we can analyze, and we'll continue to do bleeding clotting studies at the conference next year. Ongoing, of course, always monitoring the literature to understand the function of the genes. We're still trying to get medical records from everybody, especially bone density assessments, DEXs, and effects of treatment in the tetrasomy population. We're still, of course, doing behavioral measures and asking about any adult onset conditions. Our wish list hasn't changed much. In fact, this is the same slide I showed last year because there's no new money to do anything on the wish list. We haven't done very many evaluations of people with 18p minus, and none of them were adults when we evaluated them. So I think that's a really important evaluation to do. Hey, Dan, Dr. Hale, could you turn off your mic? Because I'm hearing feedback, which probably, can you mute yourself for just a second? I'll let you talk in a minute. Okay, maybe that was the source of the feedback. I don't know. Proximal 18q deletions, we evaluated just a handful, and now we have maybe 20. We'd like to do those evaluations to understand that condition better. We have evaluated half a dozen people who are in the 18q minus reference group. Now we have about 15 or 20 that we'd like to evaluate. There are people with unique deletions that nobody else has that would help us understand the genes in those regions. We'd like to do that. And of course, small duplications of any part of the chromosome. We would love to do evaluations of people with small duplications. And we actually had that, the molecular analysis tried to get funded as a GoFundMe project, got not enough money submitted that way or donated that way to even do one. So that didn't work. So we're always looking for ways to get this funded. Again, additional information is on the Chromosome 18 registry website. Also, last year we did individual syndrome research updates. There have been no big developments in the last year, so there's not much point in redoing those. So those are still on the website for you to see. And as always, if you have questions, go through Annis, and I'm going to actually let Dr. Hale talk when there's like 3 minutes left. Sorry.
Well,
Chalk it up to technology.
Everyone, I have now discovered the ultimate way to not have to give a talk, which is to be totally inept when it comes to all things electronic. But anyway, I apologize for missing the most of this while we were trying to get me on. I think we figured it out now, which means I have to work only on my laptop and not use my other screens. But anyway, hi everybody. I'm sure Janine did a better job than I did on the first half of the talk. So I don't have anything profound to say. I wish we were all someplace like, I don't know.
Yeah. Any place
together, and then we could all go out and have margaritas.
Yeah. We'll do that next year. We'll have to do double duty next year.
Right. Don't tell Shelley.
So are there questions? Yay. Yeah. We know Mariana will be here. Yay. We know that.
I will go ahead and read some questions out. Thanks for joining us, Dr. Hale. Thanks for that update, Dr. Cody. The first question that came in was from Rebecca T. And she's interested in what novel therapies are suggested for tetrasomy 18p.
Novel means it hasn't been invented yet, I guess. Mostly, should I let you talk, Dr. Hale? Regarding the bone density?
I mean, I think that I think the issue, that's not my feedback because my cell phone's not on. No, I mean, I think that there certainly are medications to deal with decreased bone density in individuals who are having fractures.
Yeah, and I think we're hoping to have people send us the information, but we think supplementation with vitamin D and calcium is actually helping the problem. So, but as people who are doing that get DEXA scans to verify that treatment effect, please send us that information. That's the only way we're going to get it, and pass it on to everybody else.
All right. Our next question, we're going to kind of go through these quickly. From Christy Kane for Dr. Cody, any new info regarding the bone density issues with tetras?
No, other than it's looking like the calcium and vitamin D help, but we just need more data from everybody. So,
got it. And for Dr. Hale, what are some things to ask our endo about concerning the bone issues? What therapies should we look in? Hardy has a newly discovered low bone quality issue after breaking his femur.
So this would not be specific guidance for that, but the general guidance is to say, as you always say, if my child didn't have tetrasomy 18p and you were presented with this picture, what would you recommend? We know that kids have low bone mineral density. There's no reason to think that they're going to have unexpected responses to conventional therapy. And there are a number. As Janine said, the initial effort is to maximize general parameters such as calcium and vitamin D, but there now are certainly medications to help stabilize bone that are widely used when people have had fractures of the spine or recurrent fractures of the extremities.
All right, thank you. And then from Camilla Downs, any further information about regressing in some areas? Lillian has developed a stutter, not ever having one before, and her phobias and fears have gotten worse. Things that she used to do, she no longer will.
Well, that's news to me on both fronts. So send us that information, the clinical reports, because I haven't heard anybody say that. And if anybody knows different, let me know. I don't remember the behavioral issues changing. In the speech, we've been concerned about potential dysarthria, but not stuttering. So, so that's new. I haven't heard that before.
Okay, got it. And then from Natalia, She said CRISPR-Cas9 does not apply to dosage sensitivity cases. Or is there a way to dose CRISPR therapy delivery, especially for duplications? Is there research on that?
Well, first of all, there are so many off-target effects for CRISPR-Cas9 that you would not, it's not appropriate for clinical use at all, period. But of course we are using it in the studies I talked about for editing cells that we can then use to understand which genes, either for duplications or deletions. So, I mean, one of my dream the experiment we're proposing is we have cells from someone with 18q- and who's mosaic. So we have cells that have 18q- and cells from them that are normal. And then we delete one gene at a time from their normal cell line and see when it starts to behave in a culture dish in the lab regarding myelination specifically. When the single gene deletion starts to behave abnormally like the 18q- deletion cells. But the converse could happen. My dream experiment is to do the same idea with someone with tetrasomy 18p, but remove one gene at a time and start to see when they become more normal. So in research, it's hugely important and totally relevant to gene dosage issues, and so it's just not appropriate for clinical use at this time.
Got it. Thank you. Well, we have run over a little bit in our session. Everyone, we currently have some syndrome breakouts going on and some other fun activities. Dr. Cody and Dr. Hale, thank you so much for all of this information. Any questions that we didn't get to today, we'll go ahead and address those in our live Q&A tomorrow. Yeah,
we'll be there for that. Okay. And also, I have a table at the table touring. Please come by, say hi. It's the only way I'll get to talk one-on-one with you. So,
and that'll be tomorrow evening. Check out the schedule when you click the reception button. Scroll down, and you'll see the schedule for today and then for tomorrow. So we hope you will go check out Dr. Cody's table and enjoy the rest of the conference. Thank you so much, Dr. Hale and Dr. Cody.
Okay.
Goodbye.
Bye.